Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem in the comment below. So today we will discuss about the DNA extraction. The first isolation of deoxyribonucleic acid or DNA from the Jonas Friedrich Mischer, which that is the scientist who first identify and extract the DNA, not identify but extract the DNA. So that give the name is deoxyribonucleic acid. So on the other hand here, basic procedure we will discuss here. Here we will discuss about the basic procedure about the isolation of DNA, and next we will also understand about the DNA sequencing mechanism and what is the benefit and what is that. So first of all, here we will take the sample collection of cells of the human, for example, which that is the desirable DNA extraction. So the cell are present in the test tube. So this test tube basically is breaking the cell membrane open to expose the DNA along with the cytoplasm within cell lysis. So the cell will be lysed or breakdown. So the detergents and surfactants used for the cell breakdown of cell wall or the breakdown of the cytoplasm. So on the other hand, it will become a mixture. So that is protease enzyme is the optional which that is used to degrade the protein or denaturation or degradation or digestion of the protein. So after the digestion of protein, that is the protein containing solution will be occur. So on the other hand, the optional RNA is used for the breakdown of the RNA for consuming the pure quality of the DNA of the desired trait. So the concentrated salt solution or saline or concentrated salt solution will add. Why? Because it become a clump of protein and lipid etc. will be clumped together, while the phenol will be added. So the phenol will be added to protein denaturation. So the denaturation of the protein will be occur, and after the centrifugation, so the centrifuge is used to separate that phase. So the DNA are present in the aqueous phase, while the cellular debris will be precipitate in the bottom of the test tube. So the ethanol propanol ice cold will be used, and sodium acetate will be used to basically produce first the ethanol or propanol ice cold will be used, and after this using the DNA also already separate due to the centrifugation. That means the sodium acetate is also used to DNA in aqueous phase will be occur. So why? Because their density is less than other protein or organelle or cytoplasm or cell membrane. So that means the cellular debris will be precipitate on the bottom. So that is the mechanism of the DNA separation. After this, basically DNA will be separate, and we can take the DNA as a sample, which that is the desirable DNA segments or DNA threads. So on the other hand, another mechanism is the solution aqueous phase containing DNA are present is here, which that is introduced with the chloroform to remove the phenol. So the phenol remove when during the phenol will be added before the centrifugation for clump of protein, lipids etc. will be formed. So that will be separate, and that phenol remove, and aqueous phase contain DNA which that is separate, and denaturation of protein is also occur by the proteinase enzyme. So that means important thing is that the chemical hydrolysis of DNA will be occur. We can add the diphenylamine indicator. Diphenylamine indicator is used for the reaction. So the heated in acid, basically acid will be add and deoxyribose sugar will be add. So this addition with the acid is basically to show the blue color by the addition of W hydroxyluvinyl aldehyde, which that is add to give a reaction in the presence of diphenylamine indicator. So that reaction will be produced and blue color by photospectrometer can detect it. So the blue color if are present, so it means the identification done of the DNA. So that's important thing is that the blue color is using for the presence of the DNA in the aqueous media. So that's here is the done of the DNA extraction. While on the other hand, next we will discuss about the DNA sequencing. After the removing of the DNA, after the extraction of the DNA and removing of the DNA, which that is DNA sequencing, we can use by here is the DNA. So this DNA as a sample used to splicing by E. coli restriction enzyme to be give DNA segmentation. So here is the DNA contain a codon, many codon, which that is contain a adenine, thymine, and cytosine and guanine, which that is the four bases are present in the form of pair. Adenine always attached with the thymine, and guanine is also always attached with the cytosine. So this after the segmentation, the DNA will be splitting into the segments. So these segments will insert into the recombinant plasmid, into the plasmid, which that become a recombinant plasmid. So this process is used by the restriction enzyme and molecular paste or ligase enzyme to paste their segment by the different process, which that is vector is come from the bacteria, which that is the extracellular DNA. And basically this DNA is come from the bacteria, and after that recombination of that plasmid will insert into the bacteria by a gene gun or another electrophoresis process. So after that recombinant plasmid will insert into the E. coli or bacteria. So after that isolation will be occur when the E. coli will be divided more and more. After the division of E. coli, it means the recombinant plasmid is also divided. It means the amplification of plasmid will be occur just like a PCR, which that is we use. So the PCR is the same mechanism which that is doing in the bacteria, which that is a natural process. After the cell division or bacterial growth to reproduce and basically asexual reproduction to produce a many recombinant plasmid, it means we will isolate. So that plasmid will be isolation. It means after the plasmid will be isolate, it means it will be more than one samples, more than two samples which that are present in a plate, and that test tube is going to incubation of DNA in plate. So during the incubation we will require the four things. First is the basis, and second is the DNA polymerase enzyme, which that is used for the polymerization of the RNA into the DNA, while the DNA is the primer. DNA primer is also used. On the other hand, one more thing is the modified DNA base or terminator is used. This modified base terminator, so the modified DNA based terminator is attached with a color inflorescence. So here is the DNA primer, which that is begin the transcript begin to make a reverse transcription And on the other hand, here the DNA polymerase enzyme is used, and modified DNA based terminator is also used. So these contain a guanine attached with a blue color, and cytosine is attached with a light blue color, as light blue color, and on the other hand, the thymine is attached with a black color. You can just imagine that, and adenine is attached with a orange color. So these color inflorescence will be shown during the segment reading, so that we will read the segment. So here is the 96 degrees Celsius temperature to treat that, and that's all mixture bases, DNA primer, DNA polymerase enzyme, and modified DNA base are mixed in the medium. So the denaturation due to the 96 degrees Celsius temperature to become again change that below the 96 degrees Celsius temperature to 50 degrees Celsius temperature to again attach the primer. So the primer will be attached and going to read and attach the bases. So these bases will be attached. After the attachment of that bases, the polymerase enzyme, DNA polymerase enzyme, will be attached last. In the presence of 60 degrees Celsius temperature, DNA polymerase will be work after the primer attachment in the 50 degrees Celsius temperature. So the 60 degrees Celsius temperature DNA polymerase use and attach the nucleotides, and these nucleotides is attachment to become a DNA. And on the other hand, the terminator base added, for example the inflorescence showing the terminator base added. So that means the addition of that terminator base to show the inflorescence of this segment. So that means the 96 degrees Celsius temperature again to repeat the more amplification. This process is done in the PCR. It means the polymerase chain reaction, because the DNA polymerase enzyme used to become a more amplification of the DNA segment of same segment. So that means the RNA is the complementary strand will be formed. It means the same daughter DNA is same like a parental DNA due to the RNA attached with the opposite nucleotide. So that means uh, I hope you make sense about that. So here's the various fragments separated, and that means the last is the terminator base added one by one. It means it show the color when it will be go into the capillary tube of the electrophoresis. So this electrophoresis contain a electrophoresis gel. This electrophoresis gel is basically the DNA segment large and lighter and heavier DNA segment which that move. It means the negative charge are present in the DNA while the positive charge are present in the end of the capillary, which that is a plate, and we will give the electricity into the electrophoresis capillary tube, and the laser are attached fixed where the movement of the segment. So the lighter segment will move first while the larger segment will move later. So that show the color. When one segment will move, it means it show the color. For example, adenine show the orange color, cytosine show the blue color, and guanine show the high blue color, and thymine show the dark black color, which that is show, and it give the sequence. It give the sequence of the color. That sequence of the color which we can give the name with the adenine. If orange color show, it means the adenine we will give the name, and cytosine and guanine, because adenine is the terminator base added, and that is attached the inflorescence color base added. So that means it show the color. So short fragments move first and longer read later or last. So the color into letters, we can this color classified into the letters. So this letter we can compare the DNA with the other, and we can study the genomic property of the human being of the DNA segment. So here is the DNA segment. So let's begin to understand the advantages of DNA sequences. So the crime recognition, disease detection, gene therapy, molecular study, and healthcare, and there are many others. So we can classify it on the basis of DNA segment, which that first go and last, which show the color, and that color will give the sequence. So this is the DNA sequencing technique. This DNA sequencing technique which gives the many benefits. So I hope you make sense about the DNA extraction process and their procedure and DNA sequencing. I had covered the two topics in the same video. So I hope you make sense about that concept. So thanks for watching. Please make sure to subscribe and bye.